Hello and welcome to the Group 38 presentation about DNA data storage. So DNA data storage aims to archive computer files into DNA molecules called nucleotides. Now these nucleotides are essentially what hold the instructions inside DNA, and they're made up of 4 different bases. And these bases are called adenine, thymine, guanine, and cytosine, and they're strung together to form DNA molecules. Now, using various computer software, such as the ADS codex, the binary code of a file to be stored can then be converted into each of these 4 bases. Now, this is primarily done by storing each bit in a file as its own base, with the zeros being translated to the A or C bases, and ones being translated into T's and G's bases. Now, artificial DNA synthesis uses the output of these bases to generate a completely unique sequence of DNA for each file, which allows for the storage of a large amount of data in a relatively small space. Now, these DNA molecules can then be stored in a tube at a stable temperature or within a bacteria that naturally duplicates. And finally, the process of DNA sequencing is used to read the information stored in a molecule, which is then translated back into binary data that a computer can read.
Now that we've covered the basics of DNA storage, I'm going to go over some of the advantages of this technology. There are many properties that were essential for developing life on Earth that make DNA a very useful structure for storing information. Since DNA is the blueprint for every aspect of life, it is important that the information stored within it is kept safe. The image on the right shows how durable the molecular structure of DNA is. Issues like flipped bits and other errors don't occur in DNA because of this structure. A second advantage that is pretty clear is the size of the structure. DNA is only about 2 nanometers thick, and therefore it can be stored in very little space. Finally, the third benefit that DNA offers is arguably its primary feature. Every cell in your body contains a copy of your DNA, so clearly it was designed to be replicated millions of times without errors. Making copies of data is very important for a data structure, and DNA can be replicated extremely efficiently with very low probability of error. Researchers are currently investigating the replication process in order to better understand how proteins travel along DNA. As the proteins travel, they zip and unzip the helix and can also make changes. With this in mind, DNA is perfect for an iterator to move through the data and make copies or edits.
There are several disadvantages to using DNA to store data, one of which is the fact that it is a very complex and slow process to store this data. In addition to this, the error rate is relatively high compared to traditional data storage techniques, and also it is very expensive, and according to Harvard, it costs up to $3,500 per megabyte to store data. And in the picture you can see the actual place where it gets slow and expensive because appending bases can take up to 1 second to do, and when these strands of DNA need to be hundreds of thousands of base pairs long, if not longer, it adds up a lot and makes it a very slow process.
As data grows, data storage will become a serious problem, and the problem will become more and more serious. This provides an opportunity for development of DNA data storage technology. The first is the storage capacity. Scientists have managed to store about 200 MB of data on DNA in 2018. The second is the diversity of stored content. The content stored on DNA include pictures, audio, PDF, and so on. And finally, because of the stability of DNA, the DNA stored in DNA is cheap and consumes very little energy. Based on these capabilities, we have to think about the future of DNA data storage.
As for the future of DNA data storage, the concept is still very much in its infancy. The primary goal currently is to address all of its disadvantages, as its theoretical advantages are reason enough to keep pursuing this new technology. The main concern regarding DNA data storage is the complexity and cost of creating these DNA strands. The current chemical-based process for synthesizing DNA is simply too costly and creates toxic waste. Instead, researchers are looking into alternative DNA synthesizing methods. Recently, in 2020, a team of researchers began looking into a more natural and efficient way to synthesize DNA through the use of an enzyme called TDT and the process known as photolithography. This utilizes natural DNA-related enzymes to do the work instead of relying on expensive waste-producing chemicals. Additionally, a new methodology of synthesizing the DNA strands as a whole is also being developed, which looks to mimic the natural DNA creation process through the use of chunks. This would theoretically speed up the synthesis process as it imitates the already refined method life relies on.
The second main disadvantage of DNA data storage is the error rate of encoding and decoding DNA. This is being addressed through two means: redundancy through the use of self-replicating bacteria to create precise backups of the DNA data, as well as more sophisticated error detection and correction algorithms. The third main disadvantage of DNA data storage is accessing specific data in DNA sequences. Previously, data files were extracted from DNA sequences by the PCR retrieval process, which searches for the particular primer that binds to the data file DNA sequence. However, this method uses enzymes, which consume most of the DNA and leads to unwanted data files to be extracted. Currently, researchers at MIT are developing a new technique that solves this issue by encapsulating and assigning each data file with a single strand of DNA. Then the researchers can extract a specific data file by adding a primer that corresponds to the matching DNA strand. This new method allows for a specified data file to be extracted without tampering with the rest of the DNA sequence.